I'm audible to you? Hello? Hello? My voice is clear?
Yes, sir.
Clear, sir.
Right. What we completed yesterday, last class?
Bryophyta markantia.
Bryophyta general character and Markantia type study.
Sir.
Isn't it?
Yes, sir.
And did you compare with Pellia?
Sir.
Did you compare the Markantia type study to Pellia? Already told you.
Same as like markantia.
Markantia fail, male, female, formation of zygote.
First of all, did you clear about Markantia type study?
Yes, sir.
What you learnt about Markantia?
Markantia general characters, asexual and sexual reproduction. Definition of Markantia occurrence.
Markantia habitat, thallus structure,
Structure.
reproduction, sporophyte structure.
Yes, sir.
And last class only I told you, after the Markantia, you have to read about Pellia and compare with Markantia type study. How many of you read about the Pellia? Please tell me the difference between Markantia and Pellia.
In Markantia, thallus midrib is absent.
Okay.
But here midrib is present.
Markantia midrib absent or present?
Present only, sir.
No one read about Pellia. Page number 173, Pellia type study.
Yes.
First of all, what you learnt about Markantia? How is Markantia thallus? Hello.
Sir,
How is Markantia thallus?
Markantia is like a green dichotomously branched moss-like structure. Dorsiventral.
Okay. Enough, turn on page number 173. Turn on page number 173 and write about external character of Pellia.
The plant body is thin.
The plant body is thin and dorsiventral. Prostate dichotomously branched.
Yes.
Somewhat wavy margin. What about Markantia? Is it similar to Markantia or not?
Markantia commonly grows in moist, cool, shady areas.
It is not similar, sir, because slightly it is similar.
What is the different character? Please respond. Hello?
Markantia.
Anthridia is present on the midrib group.
Gamma developed in new dermatophytes.
First of all, actually, you read just now the only single point about the Pelia. I'm talking about only that single point only, not entire difference.
Stomach is absent.
First of all, what you read just now, the plant body is thin, dorsiventral, prostate, dichotomously branched.
Sir.
Is it this character similar to Markantia or not?
Yes, sir.
Similar.
Similar only.
So these are the similar character, and dorsal surface is smooth with thick at the midrib. The thallus terminates to notches where the growing point is situated. These are all characters same in Markantia. Markantia also midrib and Veli also midrib. And Markantia ending notches growing point. Here also growing points are there. And the same like Markantia, the ventral surface, rhizoids are erased. But here remember, the ventral surface, the only smooth unicellular rhizoids are present. Right? And apart from the rhizoids, in Markantia, scales also present. Scales.
Yes, sir.
And do you remember the two types of rhizoids in Markantia? What are the two types? Please read once again after the class. And compare with
Wall
next
and
time.
pegged wall.
Right.
Wall and
Smooth and pegged. And scales also present. But only one difference in external morphology when compared with Markantia to Pelia, that is one type of rhizoids present, only smooth wall, and after both scales and pegged rhizoids both are absent. The scales, pegged rhizoids both present in Markantia. That is the only different, and the rest of the all are similar to Markantia. Okay? Right? Hello?
Sir, okay, sir.
Next. Next, reproduction. What is reproduction?
Production of new offsprings. Fusion of male and female gametes to form the
Enough. How the formation of male and female gametes?
male gametes are produced from the antheridia and female gametes are produced in the
Enough. How the development of Methridia, Antheridia and Archegonia, or male gametangium or female gametangium?
Antheridia is present on the surface of the callus, present in the antheridia. It is a club-shaped structure in that the antheridia is present.
Okay.
Archegonia branch is present on the dorsal side, which is above from the antheridia, in which the egg cell will produce.
Right. As you said, this first of all, the reproduction male and female gamete fusion. You said male gamete Clostridium, female gamete inside Archegonium as well. And these Antheridia and Archegonia develop from the thallus
Thallus.
at the dorsal side.
Dorsal side of the thallus.
Right?
Yeah.
Remember, these are exactly at the midrib position of the dorsal side. But what is the difference between the formation of Archegonia and Antheridia? What are the difference between Markantia and Pelia? How the formation of Antheridia and Archegonia both in Markantia?
In Markantia, Antheridia and Archegonia are present on the dorsal surface only, but Antheridia is attached to the surface, but Archegonia is a little above, flower shape, which is erect in nature. It is some of.
Any special branch in Markantia to formation of Antheridia?
No special branch is there.
In Markantia or Pelia?
In Pelia, special branch is there, sir.
Did you listen? Did you attend the first class Markantia?
Yes, sir.
Just recall once.
క్లాస్ అటెండ్ అయితే ఇది అర్థమవుతుంది. మొన్న అటెండ్ కాలేదు.
Sir, both are present on the same branch.
దాని లైఫ్ సైకిల్ అర్థమైతేనే దీని లైఫ్ సైకిల్ అర్థమవుతుంది. ఫస్ట్ క్లాస్ బాగుంది.
Right, formation of Antheridia, Archegonia in Markantia.
Antheridial branch, the formation of
Right.
Antheridia is present on the Antheridial branch.
Antheridial branch, Archegonial branch.
Yes, sir.
These are called as Antheridia 4 and Archegonia 4, right?
Yes, sir.
What about Pelia?
Pelia also Antheridial branch is there, sir.
Do you have book now?
Yes, sir. Antheridia is present in the grooves. Groove
Turn on page number 175 first. Show me where is Antheridial branch. Please listen carefully.
Antheridia are born on the dorsal surface of the thallus along the midrib.
Right, Antheridia are born on the dorsal surface on the thallus along the midrib inside the cavity. Where is branch? Where is Antheridia 4? Hello?
Sir.
Where is Antheridia 4?
No, Antheridia 4.
Don't get confused with both words, Antheridia and Antheridia 4. Antheridia is the male sex organ.
Sir.
Antheridia 4, which is the branch, the special branch to produce Antheridia. That means if the Antheridia produced one specialized branch, the branch name called as Antheridia 4. That's it.
But here no branch, separate branch.
Yes, in Pelia, no branch, no special branch.
It is present
Directly Antheridia developed on the dorsal side of the cavity.
Yes, sir. Single Antheridium.
You can observe here the turn on page number 176, diagram A.
Yes, sir.
Diagram A clearly given. Where is Antheridium?
Cavity.
Inside the cavity. Where is Archegonium? Archegonium, these both are protected with involucre.
Involucre. Yes, sir.
Right?
Yes, sir.
So now
Antheridium and
Antheridium clear?
Archegonium are protected by involucre.
Right. Now tell me the structure of the Antheridium.
Antheridium is a round club-shaped structure in which has a stalk and antheridial jacket is there.
Okay.
The cells present in antheridium is called androcytes. Antheridial jacket is a single cell layer which is inside androcytes are present.
Okay.
And stalk is there at one end. Stalk has two cells.
Simple. Already given your development. Development actually not a important, but development of Antheridia, structure of Antheridia common to all type study. Don't bother about it.
Okay.
If you learn from one type study development, you can apply for all studies of Bryophyta. The structure also same. There are the difference only in formation of Antheridia, whether it is special branch or in cavity or embedded inside the thallus. That is the different on the based on the species, right?
Sir.
So anyway, development for single cell, double cell, 4 octant stage, so like that, finally form the full-fledged Antheridium. If you take the Antheridium, I already told you 2 parts, stalk and oval body. Multicellular stalk and multicellular oval body. And this oval body protected with jacket layer. Right?
Yes, sir.
How the development of male gametes inside the Antheridium? First of all, androgonial cells. In this, initially, androgonial cells. Next, androcyte. Remember these words. Androcyte mother cell. Androcyte mother cell. Androcytes. Androcytes. Now these androcytes developed into male gametes.
Biflagellate antheridium.
Right. Especially biflagellated anthrozoids or male gametes are developed. So they are the steps. They are the steps. First of all, blue color androgonial cell. Red color androcyte mother cell. Green color androcyte. And red male gametes. So they are the steps, remember. Is it clear? You will have to written in life cycle these words. Please remember these words. Hello.
Sir.
Okay, you know Antheridium produces male gametes, right?
Yes, sir.
But how the male gametes are produced in steps? In orderly, androgonial cells, androcyte mother cell, next androcytes. These androcytes further develop into male gametes or anthrozoids. Is it clear?
Sir.
Okay.
Yes, sir.
Yes, sir.
After the class, you will send a life cycle in group.
Okay, sir.
Right. Next one. So one end completed. Antheridia one end completed. And next end, what is the next one? Another reproductive part.
Archegonia.
Right. Tell me about general information about Archegonia.
Archegonia is having neck canal cells, ventral canal cells, and egg cells.
Just wait.
Archegonia is a flask shaped.
Right. Flask shape.
It is having 3 different parts: neck, venter,
Right.
and egg cell.
Right. First of all, any Archegonia
It's a flask shape.
flask shape. Flask shape. These are the Archegonia, right? Protected in. This is involucre, this is involucre. These are the Archegonia. Drawn here only one Archegonia structure. Only one Archegonia structure. Actually, the Archegonia are formed in group of 4 to 12 at the anterior end of the branch. Anterior end of the branch. This is the anterior end. End of the branch. How many Archegonia appeared here? How many Archegonia? We can see here the 3 Archegonia. But according to the genus and species, please be silent. Hello. According to this some of the Pelia species, the Archegonia are formed in group 4 to 12. 4 to 12 at anterior end. So anyway, I pick up only one Archegonia here. Already you said the Archegonia flask shape. And now tell me what are the parts of the Archegonia?
Neck canal cell, ventral canal cell.
First of all, for outline.
Sterile
First of all, it is
outer layer is
differentiated into 4 parts. What are those 4?
Superficial cells.
stalk, sir.
Okay.
Stalk, neck,
Just wait.
stalk,
Stalk.
venter,
Right. Venter.
and neck canal cell.
Neck. Okay. And neck covered with?
Cover cells.
Right. Cover cells or lid cells?
Lid cells.
Now tell me what in interior part, inside.
Inside,
What are present inside the venter? This swollen venter.
swollen venter, egg is present.
Right. Egg is present. And
Above
Neck
that,
canal cells.
venter canal cells are present.
venter canal cell also present. And next,
Above that, 4 neck
canal
canal
cells.
cells are present.
neck canal cells also. This is the variation in one species to another species. The neck canal cells may be 4 to 14. How many of you have given Archegonia neck canal cells?
6 cells, sir.
6 to 8.
6 cells.
6 to 8 neck canal cells. Right?
Yes, sir.
Right? Okay.
Yes, sir.
Now, Antheridia structure formation clear and Archegonia structure formation clear.
Yes, sir.
What is next?
Reproduction.
Fertilization.
Fertilization.
What is next?
Fertilization.
Fertilization. What happened before fertilization? What are the changes?
Antherozoids stream to.
Please interact each and everyone.
Antherozoids will releases into the water and reaches to the
Okay,
neck
from
by
the matured antheridium, male gametes or antherozoids released out after breakdown of the wall of the antheridium.
Yes, sir.
Right. What happened meantime in Archegonia?
These are mucilaginous substance
Our
produced
cells
by
will rupture
Right.
and releases
mucilaginous
some
substance
chemical.
produced by distillation of the neck canal cells.
Right. Meantime,
ventral
these
canal
venter canal cells
chemical
and this.
substances.
Neck
Okay,
canal
right.
cells
Sugar-like.
release mucilaginous substance,
Right.
which is sugar-like substance.
These
And
cells,
one of the
venter,
antherozoid reaches the egg
right.
and fuses with the egg to form a zygote.
Right. So meantime, these ventral canal cells and neck canal cells dissolved and form chemical substances mucilage. This is exert out. Due to the exert pressure, these cover cells separated and these chemicals released out. And due to these chemicals already released, male gametes attracted. Now reach the archegonium and travel through the neck and finally reach the egg and fuse with it. This is called fertilization. What happened after fertilization?
The zygote secretes a ball and begins to increase size. Zygote formation takes place.
Right?
Zygote develops to increase its size, and cell wall vector grows and develops into calyptra, which cause the end sporangium.
The main first of single cell zygote redivide mitotically and forms the embryo. While the embryo differentiated into sporophyte, now the calyptra, those are all formed.
Okay.
Pericytium layer also. Right? First of all, this fertilization also clear?
Yes, sir.
Right. Now, so anyway, after fertilization, zygote developed into embryo differentiated into sporophyte. What are the parts of sporophyte? Already told you.
Hasturium suspensor. What about suspensor and hasturium?
What?
Suspensor and hasturium. That is hypobasal cell does not involve in division. That is zygote divides to form upper epibasal and lower hypobasal. No. What about suspensor?
Suspensor and haustorial cells. This both?
Both are same. Huh?
No. Haustorial cells only absorb the food material from the gametophyte.
What about suspensor? So suspensor activity also same as hasturium.
Suspensor activity also same, but actually suspensor only for the fixation, but not absorption.
Okay.
Okay. Where the both words given? Sporophyte development.
Yes, sir.
Where these words?
In sporophyte development only, sir.
In sporophyte development. Remember, no need to write about sporophyte development. Only we need the sporophyte structure important. Okay. But it is not important. But anyone, the sporophyte development, archegonia development, antheridia development, almost all same in each and every type study. Okay. No changes. Same hypobasal, same epibasal, from quadrant stage, octant stage, amphithecium, endothecium. Okay.
What about dehiscence of capsule, sir?
Dehiscence of capsule. Dehiscence of capsule also same. After matured, the seta elongates and rupture the calyptra and carry the capsule in the air.
Okay.
Any sporophyte capsule wall split. Split actually longitudinally split.
Longitudinally. Okay.
Yes. Right. So dehiscency also same in all type study.
Okay.
Right. Here remember only changes in structurally. Markantia sporophyte, Pelia sporophyte, Anthucina sporophyte, Phenaria sporophyte, Sphagnum sporophyte. They are all formation same, development same. Of course, maybe somewhat little 1 or 2% difference. But
in
structural,
exam, they will ask only one organism, one example.
one example. And in
Describe about
exam,
characters of Pelia means you can write only Pelia.
right? In examination, there is a chance to ask like a write about Pelia,
Okay.
general characters, Pelia life cycle, Markansia life cycle.
Pelia and
Okay.
Markantia.
I'm giving example.
Okay.
I don't know what type studies they will ask. Okay, right?
Okay.
So dehiscency after breakage of the capsule wall, these spores are exposed out. Right?
Okay.
So
Oh.
before that, let me complete the structure of sporophyte. Remember the development almost same in each and every type study. Same zygote, first two cells, epibasal, hypobasal, and later quadrant stage, octant stage, like this. So there is not much important here. The only important in structure of sporophyte. Now tell me the structure of sporophyte. Just compare with Markansia. What do you know about Markansia sporophyte?
Sporophyte is having three different parts: foot, seta, and capsule.
Okay.
Foot is attaches to the thallus,
Hmm.
and seta is attaches the foot and capsule. Seta will supply the food and nutrition to the capsule.
Okay.
In the capsule, spores are present and also elaters.
Hmm.
Present. After maturity, the elaters will help in the dehiscence of the spore, dehiscence of the capsule jacket, and the spores will releases. Spores will releases out,
Okay, released out.
and then they are germinate into the new gametophyte.
That's it. Right. I will show the diagrams here. You have to tell the difference between Markansia and Pelia. Are you able to see?
Yes, sir.
B Markansia, C Pelia. Just focus on these two diagrams. Is it clear?
Yes, sir.
Right. Only two. These two enough. No. So these two foot, seta. Any difference in seta, Pelia, Markansia?
Seta is
Here seta given.
elongated in Pelia.
Right. Seta somewhat elongated in Pelia, isn't it?
Yes, sir.
Right. The next capsule.
Capsule is smaller when compared to the Markantia.
That's not important one. Okay. No problem. Right.
Elaters are present at the
Markantia.
elaters are like bunch of elaters are represented at the same point of
Elaters are present in electrophore.
So this bunch of elaters called as elatophore.
Yes, sir.
Okay.
Of course, elaters also present and elatophores also present in Pelia. What about Markansia? Markansia only elater present. Elater present. But in Pelia, in base of the capsule, base here, remember. See here. like this.
Elater 4.
Elaters are present. This group is called elatophore.
Elatophores.
So these elatophores absent in Markansia, right?
Yes, sir.
Okay.
Elaters are present, but elatophores
Next, any
are
protection
absent, no,
layer,
sir?
Markansia,
Markan.
elatophores.
No protection is there.
Are you talking about Pelli or Markansia?
Calyptra.
Markan.
Markansia, elatophore absent. You can observe in diagram also.
Markansia, elatophore absent, but elaters are there, no, sir? Markansia, elaters are there, but elatophores are not there.
Yes. In what about Pelia?
Celia, elatophores are there.
Yes, elatophores are there. That's it. At the base, like a bunch, elatophores are there.
Group of elaters are elatophores, but there is
Right.
no groups are present in Markansia.
Right. And next,
Calyptra is present.
Calyptra present in both sporophyte. Markansia covered with Calyptra
And
Pelia also covered with Calyptra. You can observe here.
but
This is Calyptra.
it's
Right. Next,
Jacket is there.
Involucre.
Double wall jacket is there
Right.
in Celia.
in Markansia, single layered jacket.
Yes,
But
sir.
in Pelia,
Double layer
remember,
jacket is
double
there.
layered jacket. And next,
Sir, double layer jacket. It is a. Where is the double layer jacket? They,
Pelia.
Pelia.
the two are different. No, one is Calyptra and another one is Involucre.
No. In this only capsule wall also double layered. Just observe here.
Calyptra, na?
In page number 178.
Yes, sir.
The jacket has two layered cells.
Yes, two layer cells except Calyptra.
Jacket is
Calyptra
Okay.
is single layer. Okay.
Right.
And spores are present inside the jacket.
Right.
Yes, sir.
And
Involucre.
it is protected with involucre.
Yes, sir.
There is no involucre in Markansia, I think.
Right. No involucre in Markansia.
Yes, sir.
Any difference?
So involucres are absent in Markansia, present in Pelia, and double layer jacket is present in Pelia, and single layer jacket is present in Markansia.
Right. Next.
And
Inside the capsules,
elaters are single, here elaters are group.
elatophores. Right.
Here capsule Calyptra is closed, sir, in Pelia.
That is not an important one. Of course, here also, first of all, initially Markansia also closed. But when matured, the Calyptra layer ruptured.
Okay.
Okay.
Sir. Okay. Sir.
Right.
At the time of maturity, here also the jacket, two-layer jacket will ruptures and the spores will come out and germinate into the new gamete. Right.
Right. During dehiscence, the capsule wall split into longitudinally. Now the spores released out to the activity of the elaters. Indirectly, elaters involved in the dispersal of the spores.
Spores.
Like a Markansia. In Markansia also, the dispersal of spores for the activity of by the activity of elaters.
Elaters.
In
Means
Pelia,
elaters wherever they should help in releasing of spores, no, sir?
yes.
Okay.
That is the function of elaters. Okay. Next, after released, each and every spore can germinate into
new gamete.
new gametophyte. Right?
Yes, sir.
Can you draw life cycle?
Yes, sir.
I will give you just 1 minute. Please draw Pellian life cycle and put in group. Do it fast. Don't see in book. Don't say in book. In book, just given only Archegonia, Antheridia. From Archegonia, direct egg. Antheridia, direct anthrozoids or male gamete. But you will have to show the complete structure of the Archegonium and complete structure and development of Antheridium as explained in class. Don't get confused. If you have any doubt, ask me. But don't open your book. Please close all of you. Please close all your books.
Antheridiogonian cells and mother cells we have to write, no sir, here? Androgonial cell and androgonial mother cell, then antherozoids and then antheridia. Sorry, antheridia. Androgonial, androgonial cell and androgonial mother cell divides in the androcytes and finally antherozoids. Antherozoids are formed by androgonial cell which become into androgonial mother cell and androcytes and then antherozoids. Is it clear,
First-
sir? Antheridia
This
is
first androgonial cells,
okay, androgonial cells
androcyte
and
mother cell,
androgonial mother cells and
right?
androcytes.
Right.
Androcytes.
Now these androcytes developed into
antherozoids.
no.
Sorry.
Right. Enough.
And androgonial cell.
Sir, check it. Sir.
Put in group. Okay. Right. Someone told send the message.
Sir, is it correct?
Who's number 595?
ఇది ఎట్లా పంపించాలి?
Pelia, okay. Anthridial, where
Branch is not there.
branch is not here.
Sorry, sir.
Just
Androgony.
anthridial.
Sir. Androgony.
Androgonial cells, androgonial mother cells.
Androgonial mother cell.
No.
Androzoa.
What about Pteridophyta?
Archegonial mother cell to archegonia, then developed into egg. Then formation, use it to form a zygote.
Please put in group
Develop.
so that I read your name. Your life cycle post in group. I will see.
Okay, sir.
Androgonial cells.
Androgonial cells, androgonial mother cell.
Androzoids.
Androgonial.
Durga.
Sir.
Order changed. Durga, okay, you written here.
Yes,
Androgonial cell, first androgonial mother cell, after that androgonial. Next, from the androgonial, anthrozoids mother cell. Anthrozoid mother cell. Next,
Androgonial mother cell.
anthrozoids. Okay, I will write.
Androzoa.
Someone send here. Bharati.
Yes, sir.
Pelia. Archegonia 4. Why have you written Anthridia 4?
No. From reader. Okay, I will remove. Then androgonial cells are first.
Where the formation of Anthridia? No one write.
Antheridium I written, sir. Androgonial cell, androgonial mother cell. It is Bharati. Androgonial cell, androgonial mother cell, androcytes, and then
I
anthrozoids.
am asking formation of Anthridia.
Formation means
Anthridia, but mean first thallus, Pelia thallus in cavity. In cavity,
antheridial cavity. No one not written. Okay, antheridial cavity. Okay, along the midrib.
Anthridial cavity, sir. ఆ తర్వాత androgonial
cell. No. Antheridial cavity, androgonial cell, androgonial mother cell, androcytes, anthrozoids.
Androcytes.
Antheridial cavity next to androgonial cell and androgonial mother cell and androcytes and anthrozoids. 4 steps it will be. Sir, androcyte mother cell.
Then
Hello?
sir,
Audible to you?
yes, sir.
Right. Please complete failure life cycle. Okay.
Yes, sir.
I want clear life cycle.
Okay.
I can,
Not a
sir.
book life cycle. Don't copy. Right.
Okay, sir.
Next. Or what is another type study? Page number 182
Anthoceros.
on unit 13. Anthoceros.
Yes, sir.
Now just compare with Markansia and Pelia. Start with external feature of Anthoceros.
Onward.
How is Anthoceros? Structure, morphological structure.
Onward.
Horns-like structures are present on the thallus.
So it is another name, hornwarts. I will tell you why we call it as hornwarts.
The lobed thallus gametophyte without regular dichotomous branch.
Okay. First of all, complete structure, morphological structure. It is a gametophytic plant body like other bryophytes. Small, dark green, dorsiventral, prostate thallus with variously divided lobes. Variously divided lobes.
Chloroplast cells.
And thallus is several layers thick, lacking definite midrib.
Yes, sir.
Ventral surface with smooth-walled rhizoids and scales. Sorry, smooth-walled rhizoids, scales and pegged rhizoids are absent. Right. Just compare with Markansia pellia. Any similarities? Hello?
Sir, smooth wall rhizoids are present in Pelia and also in Anthoceros.
Almost it is look like resemble to the pellia.
Pelia.
Pellia also same from the lower side. Only smooth walled rhizoids arised. Pegged and scales absent. In Anthoceros also smooth and peg walled rhizoids absent.
Yes, sir.
And pellia shows dichotomously branched.
But here some
but much branched,
lobed.
much lobed, much branched thallus. And in midrib clear in Markansia and Pelia.
But here no.
But an Anthocera's midrib not clear.
Yes, sir.
Right?
Yes, sir.
So almost this is also similar to the Markansia and Pelia, right?
Yes, sir.
And after the external structure, next one. Reproduction, same copy paste with Pelia. Same like Pelia. Here in Anthoceras also, this Anthridium developed in cavity, which is embedded inside the thallus. Cavity embedded inside the thallus, and the Anthridium structure same. Right?
Yes, sir.
Anthridia structure same. What is the structure of Anthridia?
It is a club-shaped structure.
Club shaped and
Club-shaped, pear-shaped.
club shape enough. Club shaped structure with two parts, stalk and oval body.
Yes, sir. Oval body. Shock and
What are present in oval body?
oh. Androzoids are present.
How the development of androzoids?
Androzoids are
Androgonial cells, androgonial mother cells, androcytes,
Right.
and
Enough.
androzoids.
Then next, what about Archegonia? Archegonia is the same developed in. Actually, Archegonia not in tip like a Pelia. This one also developed inside the thallus.
inside.
That means here Archegonium also embedded inside the thallus.
Some are super. Okay.
Embedded inside the thallus.
Yes, sir. Embedded inside, not visible.
And we forgot our one difference in regarding to thallus.
Gemme have been found in development.
Right, gem also found. So one character forgot. Let me complete that one regarding to the thallus. Anthoceras thallus, another important one. Inside the thallus, some algal members show symbiotic nature, such as nostoc.
Blue-green algae.
Right.
Blue-green.
That is the special
Yes, sir.
feature, the special character of Anthoceras.
Yes, sir.
This character absent in Markansia and Pelia. Remember. So Anthoceras almost all similarities to Markansia and Pelia. But one special character is inside the thallus, nostoc colonies are present.
Yes, sir.
Right? Remember it. And in reproduction, in term of reproduction, no difference.
Yes, sir. Yes, same as like Markansia Pelia.
Right. Now tell me, now Anthridia completed structure. Now tell me Archegonia structure.
Archegonia inside the flask-shaped Archegonia,
Yes.
which is having 4 different parts: stalk, venter, neck, and cover cells.
Okay.
Archegonia inside the venter, egg will develops.
Okay.
Then after maturation of the androzoid mother cells, androzoids will comes out, and one of the androzoid reaches to the venter. Before that, before that, cover slips will ruptures, and some mucilage cover cells will ruptures due to the some chemical substances. The androzoid will come and contact with the egg, fuse it to form a zygote. The zygote is furtherly developed into embryo. Embryo is then developed into a spore mother cell.
No. Embryo differentiated into sporophyte.
Sporophyte.
Right. Good. Excellent.
And
Now the sporophyte structure. Right. Already told you here the variation in sporophyte structure.
yes, sir. Sporophyte
Development also same. almost same development, right?
Yes, sir. Here foot Seta is where is the capsule?
Foot okay. Sita present or absent?
Formed by an
Seta is not there, sir.
Right, Sita absent.
Seta
Remember.
absent.
And next one.
Capsule also not visible.
Right.
Spore mother cells
Sita
only.
capsule present. Just wait. Just wait. Let me complete. This one. Downloads. Okay, right. Right. Are you able to see?
Yes, sir.
Hello?
Sir.
Right. So, sporophytic structure of Anthracis, only food is there, like other sporophyte, Markansia pellia. This place, Sita place replaced with meristematic tissue.
Yes, sir.
And rest of the all as is common. Next, what is the next? Food.
Capsule.
Actually, Sita replaced meristematic tissue. Next one.
Capsule.
Capsule. This entire portion. This entire aggregated portion capsule. This entire portion called as capsule. Okay.
Okay.
From this region to top, this entire region called as capsule. Right?
Okay.
Function of food fixation, absorption. This food completely embedded inside the thallus cells. Then due to the activity of meristematic cells, meristematic tissue, the capsule structure rapidly increased, gradually increased, right? Let's see the capsule structure. And in orderly, the capsule first outer layer,
Involute
this one,
is capsule
this
wall.
outer layer, involucre is the protection layer. I will explain later. So first epidermis,
Okay. Epidermis.
right? After
Yes.
epidermis, what is the next layer?
Cortical.
What are the two cells?
Spore member cells are there.
Chlorogenous tissue.
Sporogenous tissue.
These two. Chlorogenous. This one. Chlorogenous. Next,
One minute.
sporogenous tissue. Spore cells, spore region.
Sporogenous tissue. Middle.
Next,
Again,
center, columella.
columella. Okay.
Right? Either side same.
Either side spores, spore mother cells.
Right? Spore tissues and chlorogenous tissue and epidermal cells.
Okay, sir.
Okay?
Okay, sir.
Should I draw this diagram clearly?
Okay, sir.
Please join with me. Foot, right? Meristematic cells tissues, right?
Yes, sir.
And rest of the portion called as completely
Foot.
Capsule.
meristematic capsule. Now let's see the capsule structure. In orderly, first capsule, epidermis, either sides.
Okay, sir.
Right? Yellow color line.
That is sporogenous. Is it sporogenous tissue? No, cellular.
Yellow color, outer layer.
Outer layer. Epidermis.
Epidermis.
Okay. Epidermis followed by sporogenous tissue.
Chlorogenous.
Epidermis layer, epidermal layer, this one, yellow color. Right, epidermis single layer, it is a production layer. Right? After that,
Sporogenous.
Sporogenous.
chlorogen, photosynthetic region.
Chlorogenous.
Chloro.
Okay, sir.
Green color, chlorogenous means consist of chlorophyll cells.
Okay, sir.
Chlorogenous.
Chlorogenous. Chlorogenous, right?
Yes, sir.
Chloro. After that, sporogenous tissue.
Next,
Let me draw
sporogenous.
with blue color. These are the spores, and also pseudo illators also present.
What
Let me draw
about
with
columella,
red
sir?
color.
What about
I
columella?
will draw. I will draw.
Okay.
So red color pseudo illators. Along with four pseudo illators also present. And center one
Medulla. Columella.
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 Right. Okay.
Okay.
Same diagrams. Another side same. Epidermal layer. After epidermal layer,
Chlorogenous layer.
just wait. Let me write.
Epidermis of the
Chlorogenous
genus.
with chlorophyll.
Chlorogenous after sporogenous.
Sporogenous.
And then columella. Columella is the
Right.
middle.
Right. Okay. Now it is involucre. Involucre also there. For protection of the sporophyte, involucre also arranged. Is it clear?
Okay.
Sporophyte structure. What is that? Now tell me what is the difference between Markansia, Pelia, Anthocera,
Theta
sporophyte?
is absent. Theta is
Like
absent.
number one, when compared with Markansia and Pelia, theta absent. And next one,
Pseudo-illators.
Medulla is present.
Illatophores are
right?
present in
Illators
Pelia.
and illatophore. Illators present in
Illators,
Markansia,
illatophores, and
illatophore in
pseudo-illators.
Pelia, and
Pseudo-illators.
pseudo-illators in
Pseudo-illators and present in
Anthoceras.
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 Next,
Sporogenous tissue is not there in Pelia
no.
and
Actually, sporogenous tissue present in all capsule.
Sir.
Markansia sporogenous
Center.
tissue present.
Chlorogenous.
Pelia also present.
Sporogenous
Sporogenous.
tissue is present either side of the that is medullary layer.
Columella.
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 Right. Okay. Okay. That's not an important one, but okay. Right. Next.
Chlorogenous tissue. Chlorogenous
Right.
layer is not present.
Chlorogenous, chlorophyllous cells. That means chloroplast containing cells are present in Anthoceras. Those cells absent in
Pelia.
Markansia and Pelia.
Pelia, Markansia. That means what is the function of chlorogenous tissue?
Chlorogenous tissue. Photosynthesis
means
and food preparation.
food preparation. Then what about Pelia and Markansia?
There is theta is there.
Absorption, they will absorb
Theta.
nutrients from the rhizoids.
Absorption food material from the
From the rhizoids.
Theta, see. Theta, sir.
From the gametophyte, sir.
right from the gametophyte.
Yes, sir.
But remember, Anthoceras depend on gametophyte for fixation from water only.
Yes, sir.
The Anthoceras sporophyte can prepare their own food material.
Okay.
That means the sporophyte partially depend on gametophyte.
Partially.
This Anthoceras sporophyte partially depend on gametophyte. Remember.
Yes, sir.
And the center position occupied by columella.
Columella means phloem. Sorry. Columella
This
is a
is completely
combination.
sterile tissue. These columella absent in Markansia and Pelia.
Yes, sir.
Yeah.
Okay.
Yes, sir.
Columella absent in Markansia and Pelia.
Yes, sir.
Right.
Yes, sir.
After dehiscence of the capsule, the spores reached out and these spores further developed into
New gametophyte.
new gametophyte.
Yes, sir.
Right.
Yes,
The
sir.
life cycle. With Antheridia structure, Argonian structure, sporophyte structure also in orderly. Actually, no one write about sporophyte.
Embryo develops into sporangium. Sir, sporangium, capsule, spore mother
Embryo,
cell.
after embryo, sporophyte. That's it. After sporophyte differentiated into food, meristemate tissue, capsule. And
We
in
can't-
capsule parts also you have to mention.
Means sporangium, we don't want to write. Sporangium, it's sporophyte.
Sporangium, nothing but capsule.
Okay. So then we write sporophyte, that's sporophyte capsule and spore mother cell.
Okay,
Finally spores. Huh?
right.
The life cycle same as reader, it is not considered as sir.
What?
Same life cycle from reader. Is it not required?
Don't say in book.
Now not in examination part.
In examination part,
Then we write like this only, no, as per reader.
that's enough.
Okay, okay. While we are preparing, we have to write same life cycle, no sir?
Same life cycle.
As
Same
in
like
reader.
me. That's why I told you. As it
No,
is.
no. As in reader.
Yes. Umarani. Complete it.
కరెక్టేనా సార్?
ఎక్కడ సెండ్ చేసినావు?
గ్రూప్ లో సెండ్ చేసాను.
గ్రూప్ అయితే నేమ్ వస్తుంది, గ్రూప్ లో రాయలేదు, సెండ్ చేయలేదు.
మీరు ఇప్పుడే మెసేజ్ పెట్టారు. దాని పైన మీకు కూడా పెట్టాను. మీకు కూడా పెట్టాను సార్.
Androcyte, mother cell, androgonium, androcytes, okay. Egg, zygote, embryo. ఏది నాకు స్పోరోఫైట్ కనిపిస్తలేదు అక్కడ.
కనిపించట్లేదు. అయ్యో.
Okay. Just see, listen all of you. Listen. Anthoceras, haploid, Antheridium, Archegonium formed inside cavity, okay? Cavity. Sir, just 5 minutes. 3 minutes, sir. Antheridium, club shaped. Stalk, body, inside body androgonial, androcyte mother, next
Androcyte mother cells.
Androcyte
Androzoids.
or male gamete.
Yes, sir.
Archegonium,
Archegonium.
stalk, venter, neck, lid cell.
Lid cells.
In venter canal cells and egg also present. Or female gamete.
Yes, sir.
Male and female gamete fused. Zygote, embryo, sporophyte, right? Diploid,
Yes.
sporophyte, food, meristematic tissue, capsule. What are the parts of the capsule? In orderly, first one.
Foot.
Capsule lo
Stalk ఉంటుంది సార్. ఫుట్ సార్.
క్యాప్సూల్ అండి, స్టాక్ ఎక్కడ ఉంటది?
ఫుట్ సార్. స్టాక్.
స్పోరోఫైట్ కాదు, క్యాప్సూల్.
4 mother cells, sir. Illators, 4 mother cells.
In order, first outer layer.
Epidermis.
Epidermis.
Chlorophyllous tissue.
Okay. Sporogenous. Next.
Tissue.
Chlorogenous. Sorry, chlorogenous.
Chlorogenous tissue.
Next.
Central columella.
Porogenous. Okay.
Porogenous and columella.
Central columella.
Polymer.
The spores after release developed into anthocerous gametophyte.
ఇదంతా
Okay.
లైఫ్ సైకిల్ లోనే ఉండాలా సార్ మొత్తం?
మొత్తం లైఫ్ సైకిల్ ఉండాలనే కదా చెప్పేది.
ఓకే సార్. అంటే
ఐ విల్ సెండ్ క్లియర్.
స్టేజెస్. ఓకే సార్.
ఏదో ఆట ఆడుకుంటాను అనుకుంటారా ఇదేమన్నా? I will send clear life cycle. Okay.
ఓకే. ఓకే సార్.
After the class, I will write and send you.
ఓకే సార్.
Please compare with your life cycle.
ఓకే సార్.
Okay.
ఓకే సార్.
Next time, don't make mistakes.
ఓకే సార్.
Right. Please join next class, Dr. Sudharan Madam.
థ్యాంక్ యూ సార్.
ఓకే సార్. థ్యాంక్ యూ.